Thanks for having me. Thanks for being here. This talk is about how to start with the wealth of connectomic data that we now have access to in the fruit fly and build models from that. And it's led by Jana Lapalainen, who's in the back over there, working with Jakob Anke, who's in the back over there. So the team is here for more questions.
Sorry, can you speak into the mic?
Yes.
Or you can move the.
Problems. Is that good?
Yes, thank
Great. Okay, so let me just introduce very quickly the star here. This is the fruit fly. It's about 2.5 millimeters large. And the cool thing is that we now know a lot about its nervous system. It has a brain that's shown here. This is a late microscopic image of it. And it has a ventral nerve cord, which is homologous to like a spinal cord. The nervous system is largely conserved across individuals, similar to C. elegans. So that means you can measure the connectome in one animal, neural activity in one animal, behavior in yet another animal, and hope that maybe the same nervous system is underlying all of these measurements. The very exciting thing that happened last year is that our understanding of the fruit fly visual system, the fruit fly nervous system, went from this to this, where now we have an electron microscopic reconstruction of nearly every neuron in this nervous system and how they're connected to each other. We know now that there's about 140,000 neurons in the brain, about 23,000 neurons in the ventral nerve cord. These numbers I bring up because it's a very small number of neurons compared to any artificial neural network that people now work with. For context, AlexNet, which I don't know how many of you heard of from over 10 years ago, has 4 times as many neurons. And all it can do is recognize 1,000 objects. And this fruit fly, with this small number of neurons, can walk, it can fly, it can look for mates, it can look for food, it can avoid predators, and many, many more things with a very small compact nervous system. So it's there's a chance that maybe because it's so small, we can understand it. Okay, here's my computational neuroscientist caricature of how the brain works. At the bottom are microscopic phenomena that this network of neurons then determine how the substrate of the nervous system works. So it's it's how is this neural network connected, the neural connectivity. It's the dynamics, the single neuron biophysics, the single synapse biophysics, the synapses that connect them, neuromodulation, synaptic plasticity. And there's probably many other phenomena that go into how the brain works that we have yet to even discover. Now together, these microscopic phenomena give rise to the neural activity. That's a collective phenomenon of this network of neurons. And the neural activity underlies behavior. And the behavior that an animal produces, usually you can think of as there's some goal for this animal, and the behavior serves to get the animal to that goal. So you can think of that as a task. So we have this hierarchy here. And the very exciting thing is that we can now measure the connectivity in great detail. And we know something about the biophysical properties of neurons and synapses. So you could ask, given these measurements, can we now predict neural activity, behavior, and maybe go up the hierarchy? And I think the answer is no. Connectomes by themselves are not sufficient, and we don't know for each neuron exactly what its biophysical properties might be. And so while we can measure a lot of detail about the structure of the neural network, it doesn't tell us anything about the precise dynamics. And so, that means that this, in terms of building a model, we're still in the under constraint regime. There's a lot that we don't yet know. So if I build this model, there's a lot of different patterns of neural activity that this circuit might generate, depending on the choices that you've made for the dynamics of the neurons. But the good news is we have a lot more measurements that we have been able to make from the brain and from the animal. We can measure neural activity, not everywhere and not all the time. We can often infer what the fly is trying to do, especially in experimental settings. And we can measure behavior. We've been able to measure animal behavior for centuries using eyeballs and now with cameras, video cameras, and neural networks. So, these extra measurements can be used to constrain our models. So let's talk about how we can do that. Now, you've all heard about neuro AI. That's a really sort of a popular modeling framework that's now in vogue. Thanks to the advances in deep learning, we can now use machine learning to train up a artificial neural network to mimic the behavior of an animal or to solve a particular task that we imagine the animal might be solving, and then inspect the artificial neural network, the trained artificial neural network, and ask how does that neural network solve the task. Now, this is also an under-constrained modeling framework because many different connectivity schemes, many different nonlinearities, single neuron dynamics can give rise to the same effective behavior. And so we know this in artificial neural networks where you could train a transformer or a convolutional neural network or an RNN to solve similar problems in computer vision. And these are very dramatically different architectures that use completely different computational mechanisms and yet can solve similar tasks. So this is a valuable way of generating hypotheses, but still quite under constraint. And so, in this work, what we asked was, what if you bring in constraints across these scales? If you know something about the task that a circuit might be trying to solve and measurements at the microscopic scale, are those sufficient? So this study, you should think of as a thought experiment where what we did was we kind of tied two arms behind our backs. We asked, let's say you only know the connectome and information that you can get from a dead brain, and you know something about the task. How well can you predict neural activity? Frequently we do have neural activity measurements, but in this case, we're pretending that the only measurements we have come from a dead brain. Okay, and then the idea is if you do this, this can kind of help us understand how much information is sort of contained, captured by the connectome. So this is kind of the workflow. We modeled the motion pathways of the fruit fly visual system. The idea is that, over the last 10 years, my colleagues at Janelia have published a series of papers mapping out the connectivity in the visual system. These connectomic measurements were used to constrain a model, to construct a model. And the exciting thing is that over the last, over a decade, we now also have a large number of studies, 26 studies, that have looked at, have made functional measurements in this same circuit. And so we can compare the neural activity predictions from our model to the neural activity measurements made in the lab and ask how well does this model constructed without any of that neural activity predict neural activity. So if we can do that well, then that suggests that we have a strategy for generating predictions of neural activity from connectivity. So the place where we looked at this was the fruit fly visual system, the motion pathways. What you see on the left is a schematic diagram of the fly visual system. The very cool thing about the fruit fly, it has two very large compound eyes, and the pixels, the ommatidia here, are arranged in a perfectly crystalline hexagonal lattice. So that's the retina over here that you see in red. The neuropil, the neural network that processes this information, and the optic lobes also have the same hexagonal lattice arrangement to their circuitry, and so there's columns that have this exact organization. And what that means is that there's neurons from columnar cell types which appear exactly once per column. So there's one of them per pixel, you can say, also arranged in this hexagonal lattice. And their conductivity is largely sort of local in a way that matches this organization. So what we knew as of maybe 5 years ago, our understanding's been dramatically transformed in the last year, was the connectivity for 64 cell types in this optic lobe. And so what you see here are the R's, the photoreceptors. All of these have names. We know a lot in the fruit fly, but don't worry about the names. So this is the connectivity between cell types for 64 cell types. We also know the signs of these synapses based on transcriptomic profiling. We know which neurotransmitters are expressed, released by each presynaptic neuron, and also some idea of the receptors on the postsynaptic side. And so using that, we were able to color code these connections into excitatory and inhibitory connections. And we also know for each pair of neurons how many synapses are made. So that count, synapse count, is reflected in the sizes of these squares that we have here. So this is an incredible amount of detail, the information that we have access to. And so you can use this to build a model. In this case, what we modeled is the right eye. So each hexagon here is an image representing the neural activity in the population of neurons of a particular cell type. So each pixel inside one of those hexagons is a single neuron. The entire hexagon is the population of neurons of that cell type. And so we have connections between cell types represented by these lines here that go between cell types according to the matrix that we showed you earlier. We also know for each pair of cell types, let's say you have MI9 and T4D, we know the spatial organization of the connectivity. And so the numbers that you're seeing here are the average connections between a neuron that's displaced spatially from this neuron in this direction, the average number of synapses that you would see in each of these directions. And so what that means is we really have much more information, not just connectivity at the scale of cell types, but actually at individual neurons. So this is a connection matrix of about 45,000 neurons. So each pixel here is a neuron and not a cell type. And this is the organization that we're actually modeling. Okay, so now that we have this, how do we model it? We need to put in some dynamics. So this is a recurrent neural network that has feed-forward connections, lateral connections, feedback. How do we model this? So we decided to use the simplest sort of model of each neuron and synapse, but keep the detailed connectivity. So the idea here was to ask, how much does the connectivity constrain the computation at the network level compared to, excuse me, the single neuron properties. So the single neurons now are simple, but even the simplest model that you can imagine here at passive point neuron has two parameters. There's a time constant that's here and the resting membrane potential. So we're modeling the voltage dynamics for this passive neuron, and that's V. And there's some synaptic input Sij. So that's where all the magic is. That's because that's where the connectome is. So the input that a neuron receives from its presynaptic partners in this model depends on the weights, of course, but also the presynaptic activity. And the way in which, in the early visual system, these are largely non-spiking neurons. So we're not modeling any spiking, but we have a simplified model of the graded release synapses, basically. So what that means is that the neurotransmitter that's released is roughly proportional to the presynaptic voltage, but there is a threshold below which there's no release. So that's a rectified linear nonlinearity that we use. That's the simplest sort of representation you can have for a graded potential synapse. Okay, so that's so the magic is all in the weights, and how is that so? Well, we know the number of synapses there are between any pair of neurons. So that tells us something about the strength, but we don't know how strong each individual synapse is between a pair of neurons. And so that's a parameter that we don't actually have access to. So that's this alpha term. That's basically the unitary strength of a single synapse. And in this model, what we've said is that that only depends on the cell type, because it should only depend really on what neurotransmitter was released at that synapse and what receptor was used to detect it. The rest of the details of the connectivity, the weights of a connection, are actually measured from the connectome. So N is the number of synapses between that pair of neurons, and sigma is a sign, which, I already told you we had some way of measuring also from a dead weight. So the parts in green come directly from the connectome, or the augmented connectome if you add in the signs. And these parameters in red are the parameters that we now need to have some way of figuring out what they are in order to simulate this circuit. And all of these parameters depend only on the cell type and not on the individual neurons themselves. And that's because we believe that these are properties that have to do with their transcriptome, and so perhaps all neurons of the same cell type share those properties. And so now with the connectome and with the cell type information, our model for about 45,000 neurons only has 734 free parameters. That's a very small number of free parameters, and the number of parameters only scales with the number of cell types. If you didn't have any of this and you had to estimate the weight matrix from scratch and all the other parameters from scratch, it would actually be more like 2 million parameters that you have to estimate for this giant network. And so there's a massive reduction in number of parameters just because we're directly measuring from the example. Okay, so now that we have this, we need to inject, as I said, what we're trying to do is to marry these microscopic measurements with a macroscopic constraint of what the function of the circuit is. And so the way we phrase this is, whatever those parameters are that we don't know, they should be good for doing vision. And in particular, these are for the motion pathways, so they should be good for doing motion vision. And so with that sort of high-level goal in mind, what we did was we trained this circuit in an end-to-end fashion to perform a computer vision task, that of detecting optic flow. And so the idea was that we took this computer vision data set that has video clips, and where for each frame in the video clip, there's an optic flow field, the ground truth optic flow field available, where for each pixel, there's a vector that says, where does that pixel move in the next frame? And so we took those videos, we rendered them with a fly-eye sort of rendering, and then we trained this whole network in an end-to-end fashion in order to be able to detect, to solve this task. We have a decoder that transforms from the representation of motion used by the fruit fly visual system into the representation used by computer vision scientists. So in that way, we don't, this is a discovery tool. We want to say, we don't really know which neurons are motion selected. We don't know how they represent motion, but somewhere in here, there needs to be a motion computation, and this allows us to do this. So we trained this whole thing using backpropagation through time. This is a system of 45,000 coupled differential equations. Fairly large network, 64 cell types, but we're able to train this whole thing end to end. So then we asked, once we did this, how well can we predict the neural activity in the circuit? And so for this, we went back to the literature. We mined the literature. We found 26 papers that made recordings in various of these cell types in this amongst these 64 cell types, and asked, if we showed the same laboratory stimuli to these neurons, what would the neural activity that they measured look like? And we simulated that and characterized it. One of the simple things you can do is to flash a single pixel on and off. So there you can either flash it up, increase the brightness, or decrease the brightness. Do it for different durations, and different labs use different protocols, and so all these things, there's no standardization necessarily there. Some measured calcium, some measured voltage, and so we simulated it all, and we looked at the scale of segregation into on and off pathways, how well we could predict that. This is one of our models, and this model gets all of this right except for this one cell type TM4. And these are neurons, these are cell types, this is a small number of cell types that are very famous, and so I'm showing those here. But we actually, sorry, let me just, and then here's another set of famous cell types. These are these T4 neurons and T5 neurons that come in 4 different flavors. These were discovered 11 years ago by Oxopor's lab and found to be elementary motion detectors, the first site of motion detection. And they are tuned to motion in 4 cardinal directions, and we're able to predict that correctly as well. We can predict the voltage traces in each of these neurons. This is the voltage trace for T4. We can also ask, what does the excitatory current look like? What does the inhibitory current look like? There's a very large degree of detail in this model, and we're predicting all of these details. We can also tell you which cell type provides what input to each of these neurons, telling you where they, shedding light on the mechanism of direction selectivity in the preferred direction versus the null direction. Okay, so what we also did was we trained 50 models, not just one model. Thank you. And here's, all 64 cell types. Only half of them have been experimentally characterized. To on and off pathways. And if you train 50 models, what we find is that most of the models, the median model, for instance, gets the prediction right across all of these cell types. So we can predict across this ensemble of models with basically 100% accuracy disaggregation into on and off pathways. Similarly, we can predict motion for on motion and off motion in T5 neurons and not in their inputs. So there's a very large number of predictions, actually. Jana's paper has a supplement with 450 pages of predictions, where for each cell type, we have, for every visual stimulus that we think about, predictions of how this ensemble of models will work. And this is actually similar to what was shown earlier. Here's across this model space, the distribution of models, and actually you find that they cluster nicely, and we're able to discover 3 clusters. There's a lot of interesting detail there that I will skip. Just to summarize here, that I think through the study, we're able to show that if you have detailed measurements of the neural activity, you can actually fill in these other bits, the single neuron and synapse biophysics, and using task constraints. And through this, we're able to predict neural activity in great detail, as well as the mechanism of the computation. So, I hope I've convinced you that with the connectivity, we can make these predictions. But if you didn't have the connectivity, and let's say you made great detailed measurements of single neuron biophysics, but didn't have the connectivity, we know from all the work in AI and neuro AI even, that just knowing the transfer functions of single neurons doesn't necessarily constrain this enough. There's many connectivities patterns that can solve a particular task, and so that's not sufficient. So this connectivity measurement is in some sense special. So with that, I'll stop. I have a few slides on whole brain modeling, but I will skip that and thank my